ఇంటర్మీడియట్ విద్యారంగంలో అగ్రగామి మన NSR ఇంపల్స్ ఎడ్యుకేషన్ ఇన్స్టిట్యూషన్. ఐఐటి జేఈఈ అయినా నీట్ అయినా తిరుగులేని విద్యా సంస్థ NSR ఇంపల్స్.
Now in plant growth and development we are discussing about growth processes. Now if you take the growth processes, what are the growth processes we studied? Differentiation. We studied the first phenomena, it's called differentiation. What is the next phenomena? De-differentiation. What is the next phenomenon? De-differentiation. The first one is differentiation, second one is de-differentiation, and the third one is, let us talk about the third one, re-differentiation. Okay, differentiation, de-differentiation, and then what we have is re-differentiation. So for the previous class we studied differentiation and de-differentiation. Guys, I told you to start with the good notes and practice all the examples. Everything I am trying to give you in the form of block diagrams. A simple block diagrams I am giving you. Make sure that you practice on the notebook. Try to complete that day class listening and practicing on the paper. Try to complete on the same day. So for the last class, whatever we discussed, differentiation, definition, you listen once, practice examples. De-differentiation, practice the definition and practice all the examples. Now we are in the third process, that is re-differentiation. So what is re-differentiation? Re-differentiation is nothing but, we can say definition, maturation, or we can define this as differentiation of de-differentiated tissue. De-differentiated tissue. Okay, I repeat again. What is the definition for re-differentiation? Maturation or differentiation of de-differentiated tissue. What's the meaning of de-differentiated tissue? You all understood what is called de-differentiation. Yes sir, you have understood what is called de-differentiation. Now what did you study in de-differentiation? Now let us talk about cambium. Cambium once again may become meristematic and starts producing new cells. Did you remember secondary growth of dicot stem, right? So here in this case, what we say, cambium divides and produces new cells. New cells what are produced here? Okay, new cells what are produced here, meaning here the meristematic cells, meristematic tissue. What is meristematic tissue? Who is undergoing the process called cell division. Now this meristematic tissue, what did you study in de-differentiation? This permanent tissue once again becomes meristematic, start dividing cells on either side. Okay, start dividing cells on either side. Now this meristematic tissue once again loses the ability to divide, and convert into and becomes permanent tissue or mature tissue. Permanent tissue or we write mature tissue. Okay. What is differentiation? Cell division, elongation, converting, formation of permanent cell, maturation, it's called differentiation. Now the permanent cells, they will once again become meristematic and dividing, it's called differentiation. Now, this meristematic tissue will lose the ability to divide, once again becomes, second time it becomes permanent or mature tissue, then that will come under redifferentiation. Now, let us talk about the examples. Now, let us see that the cambium when it divides and stops dividing. So, this is converts into secondary phloem and this converts into secondary xylem. Okay, this converts into secondary phloem, this converts into what is called secondary xylem. Secondary phloem, secondary xylem. That means formation of secondary phloem and secondary xylem from where? From cambium, right, and then periderm, right, and then periderm formation. Periderm formation. Now, this will come for what? Redifferentiation. I repeat, what is redifferentiation? Maturation or differentiation of dedifferentiated tissue. What are they? Meristematic tissue undergoes cell division, loses the ability to divide and becomes permanent or mature tissue. When meristematic once again loses the ability, once again second time converts into permanent or mature tissue, this is called redifferentiation. What is example? Cambium dividing actively. These all are meristematic cells what are produced here. These once again they lose the ability, structurally, functionally they are differentiated. One into secondary phloem, other into secondary xylem. Later, at the end of secondary growth, even you can see formation of periderm. So, formation of these three are the examples for redifferentiation. Okay. So, let us study all together the examples for all three processes. I hope you understood this redifferentiation. Examples. Let us talk about examples. Now, if you take secondary growth, so you can take example dicot stem. What happens in dicot stem? Initially procambium. Procambium undergo the process called differentiation and produces what? It will undergo primary growth. What it produces? Primary phloem and primary xylem. This formation is a good example for what? Differentiation. Yes or no? Now, next what happens? Now, these are permanent cells. These are permanent cells. When you take this is a permanent cell. Later what happens? This is dicot stem. Later what happens? Taking the help of medullary cells, taking the help of medullary rays, what happens? Now these will undergo division, extend, medullary rays also will undergo division and form what? What is that? What they form? They form what is called as interfascicular cambium. So formation here up to here is differentiation. This is differentiation. When the cells divide and continue, here also cells divide and continue, they form this cambial ring. Yes or no? And this is intrafascicular cambium, and this is, what do you call this? Intrafascicular cambium. You remember you studied in your dicot stem anatomy, secondary growth. Intra is primary differentiation. Inter is interfascicular cambium is formed as a result of, this is first differentiation, this is second one called de-differentiation. This is called de-differentiation. Okay. Formation of primary phloem and xylem is called differentiation. Formation of interfascicular cambium is by de-differentiation, then followed by. So here already primary phloem is there, here primary xylem is there. Further what happens? Cambium undergoes cell division, lead to formation of the cells. So formation of secondary phloem, formation of secondary xylem, both will occur. Okay, both will occur. Now this will come under secondary growth, that happens by redifferentiation. Are you getting me? Dicot stem, if you take example, in the primary growth time, when young stem is growing, procambium divides and forms primary phloem, primary xylem. That's a good example for differentiation. You know story what happens later. Later intra is in between here, but intrafascicular cambium cells extend and divide laterally. Here also they divide. Medullary cells also divide. They form what is called here, what they form, it's called interfascicular cambium. This is for de-differentiation. And later the secondary growth ending, cambium once again becomes meristematic and divides the cells, and these cells once again they lose the capacity to undergo cell division, converted into permanent cells. These permanent cells are secondary phloem, secondary xylem, and even later there is a formation of periderm. All these things are examples for redifferentiation. So I am giving you one example, dicot stem. Another example I will give you, tissue culture. Another example what I will give you here is tissue culture. What happens in tissue culture? The first one cell what I take in the tissue culture in the test tube is one cell. What do I call that? Explant. Okay, or I call stem cell. Yes or no? Stem cell will undergo cell division. So at the time of cell division, this itself is called differentiation. It undergoes differentiation and forms a group of cells. This group of cells are called pith cells. Okay, so it will undergo cell division and forms a group of cells. What do you call this group of cells? This group of cells produced are called pith cells. You know medullary cells. In anatomy part you know medullary cells. Pith cells we call. Right. These pith cell further undergoes de-differentiation. As a result lead to formation of callus. So pith cells are get converted into callus. Callus formation occurs by de-differentiation. Then followed by what occurs here, it's called redifferentiation. I hope it is not clear to you. I don't think it's visible to you. Again I'll write. Okay, these they undergo de-differentiation and forms what are called callus. You know what is callus? Undifferentiated mass of cells is called callus. That is how it is produced, by de-differentiation. It further undergoes redifferentiation and lead to formation of root apical meristem and shoot apical meristem. That means formation of root and formation of shoot is by the process called redifferentiation. I hope now it is very clear. Let us take two important examples having all three processes. Dicot stem, if you take, the very beginning procambium is there. Procambium will undergo differentiation, that is at the time of primary growth leads to formation of primary phloem, primary xylem. That is the process called differentiation. Later what happens? Cambium at the time of secondary growth extends laterally. What extends? Intrafascicular cambium extends laterally and forms what is called interfascicular cambium, that occurs by de-differentiation. Later this continuously forms the cambium ring. Now due to activity of the cambial ring, cambium once again becomes meristematic, starts producing new cells on either side. These new cells again they lose the ability to undergo cell division, converts into permanent tissue called secondary phloem and xylem. Later they form periderm, that is because of redifferentiation. This is a three processes what you can see where in dicot stem. Coming to tissue culture, single cell what we take, it's called explant. Explant is nothing but single cell. It undergo differentiation and forms group of cells called simple parenchymatous cells. They are called pith cells or medulla cells. And later they undergo de-differentiation and forms callus. Callus is undifferentiated mass of cells. And then it undergo redifferentiation, forms root and shoot, that is root apical meristem and shoot apical meristem. These are the good examples for all three processes: differentiation, redifferentiation and redifferentiation. All three are very, very important. Examples you practice on the paper. Remember these two examples with three processes thoroughly. Now what is called development? Now let us talk about a small process, but you know we studied about growth processes. Growth processes are three: differentiation, de-differentiation, redifferentiation. Further continue, let us see what is meant by development. So let us write the definition, listen carefully. Development is defined as the process of or sum of processes, growth and differentiation. So far we discussed about the growth, we discussed about differentiation. Sum of growth and differentiation, it's called development. Sum of growth and differentiation, it's called development. Or it is also defined as, you can also take one more definition. What is the other definition? The process includes starting from seed germination up to senescence. That means from what you can write, the stages of life cycle, better you write down only, stages of life cycle. Stages of life cycle in higher plants. Okay, what is nothing but development? Development is referred as stages of life cycle in higher plants from where to where, starting from seed germination up to senescence. Seed germination means from the day one when seed started germinating into a young plantlet until it undergo death and decay process, until the seed grows, it becomes an adult plant, it gives fruits, flowers, vegetables, whatever we get, and it will undergo death process. So, complete lifespan starting from seed germination until it undergoes senescence or death. Whatever the stages which happen, whatever the changes they take place, everything come under development. What is development? Stages of life cycle in higher plants from seed germination up to senescence is called development, right. So, if you take the first seed germination, if you took, now it will undergo meristematic cell. What is that meristematic cell? It will go cell division, then cell elongation. What happens at the time of cell elongation? Protoplasmic growth occurs. In each cell. At the time of seed germination, meristematic cells keep undergoing cell division. At the time of cell elongation, inside the living content called protoplast, protoplast is increasing in size, that's called protoplasmic growth, and then followed by cell differentiation. As a result of cell differentiation, we can see formation of mature cell or a permanent cell. Then the plant grow, it becomes an adult grow plant and it attains what is called vegetative phase. When the plant becomes adult, after cell division, cell elongation, cell differentiation, then there is a formation of mature cells and permanent cells. The plant is attaining what is called growth phenomena, and after growing, the plant becomes an adult plant. Adult plant shows vegetative phase or juvenile phase. Vegetative phase means it forms complete root, stem, leaves and all that. Then it enters into reproductive phase. Reproductive phase, it undergo reproduction, transfer of pollen grains from anther to stigma, maybe self pollination, cross pollination. As a result, you can see again formation of young plantlet, right, that is happens by seed germination, and this will undergo what is called death or senescence, or what is called senescence. Okay, after the seed germination, after seed is produced, right, then the plant will undergo death and senescence. The life cycle starts with the seed germination. Meristematic cells keep undergoing dividing, elongation, differentiation, becomes an adult plant where the development of vegetative organs, develops into reproductive phase, produces seed and finally leads to death or senescence. Now, in this development, let us take an example of development. So, I am writing here development. Let us study example for development is plasticity. Okay, one of the example, you can take any change as development, any change you can take as development. Let us take one of the example for development processes, the process of plasticity. What is the meaning of plasticity here? Plasticity here, it refers, I am giving you definition here, make a note of it. The process of formation of a different plant parts in response to environment, and follow, you can write here different structures, better word, different structures in response to environment and form different pathways I repeat again. What's a development? One of the example for development is plasticity. What is plasticity? The process of formation of different structures. Okay, what is plasticity? The process of formation of different structures. Plasticity meaning process of formation of different structures. Within the plant, you can see formation of different structures in response to environment. According to the surrounding environment, they form different pathways. It's called plasticity. With the definition, you don't understand anything. Let me give you an example, then you will understand. So, what is this development? Sum of growth and differentiation. Starting from the life cycle of the plant in higher plants, starting from seed germination to senescence is called development. So, this is how life cycle begins. This is called development. Now, we are in the discussion of example of development is plasticity. So, what we are discussing here? We are discussing plasticity. What is plasticity? Example for development. What is a plasticity here? A process of formation of different structures in response to environment and forms different pathways. So, here you can take, for the plasticity you can take one more example for plasticity is heterophily. Okay. For plasticity, what is the example? Heterophily. What is heterophily? Let us take examples here. Example, let us talk examples for heterophily. So, here you can take cotton, coriander and larkspur. What happens? Why do you call this as heterophily? In this cotton, coriander and larkspur, we find a different structure of leaves. Different structure of leaves. The formation of different structure of leaves is heterophily. So, here for plasticity we can take plasticity in terms of leaf. If you can take, it's an example called heterophily. For heterophily examples are cotton, coriander, larkspur. Hetero, different structures. Phily means leaf. In bryophytes you remember you studied rhizoids, root like, colloid, stem like, phylloid, leaf like, phily, phylloid, phyllotaxy, alternate, opposite, what you call spiral, alternate, opposite and whorl phyllotaxy. Phyli, phyllotaxy, phyli, phylloid. Wherever you get phyli, we talk about leaves. Phyli means leaves. Hetero means different structures of leaves. Where do you find? You find in case of cotton, coriander and larkspur. What are the different structures you form? So what you can see here, so can you stay 5 minutes? So here if you look at, so let us take the example coriander. In case of coriander, if you see the young plant has only 2 lobes. I mean young leaf has only 2 lobes. The structure is different. When young leaf, when it becomes the adult leaf, then it can see so many lobes. That means young leaf is different from adult leaf. Adult leaf is totally different in structure from the young leaf. Adult leaf shows many different lobes when compared with the young leaf. So here different structures of leaf is a very good example of heterophily. Heterophily is a type of plasticity. Plasticity is one of the developmental process. Now let us take plasticity definition. Process of formation of different structures. Okay, here you can see process of formation of different structures. In young leaf one structure, in adult leaf another structure. And in response to environment to form different pathways. What's the meaning of environment here? You can take one more example, that is environmental heterophily. What is that? Environmental heterophily. In environmental heterophily, you can take the example. What example we can take in case of environmental heterophily? You can take example called buttercup. So this is nothing but CUP cup. Okay, so what is environmental heterophily? The best example is buttercup. What happens in case of buttercup? If you take the water, in this case here, the plant what you find here, aerial leaves are broader, occupies more space. The leaves what are present inside the water, they are like ribbon shape. So here aerial leaves, what you find here, they are broader, and underground leaves, or you can say submerged leaves, are ribbon shape. What you can see here is aerial leaves are broader. What differs here? Here the environment is air. Here the environment is air. Here the environment is water. That means if you take the buttercup, half of the water is aerial, above the water level, it's completely aerial. We can say aerial. So half of the plant is submerged in water. Half of the plant is aerial, half of the plant is submerged in water. So aerial leaves, if you take, aerial leaves are completely developed, they are broader, they are completely broader and they completely occupy some surface area. On the same plant, if you look at the leaves which are formed inside the water, half plant in the air, half plant aerial, half of the plant is submerged in water, half of the plant is submerged in water. That means here in water you can see submerged leaves. Submerged leaves are nothing but ribbon shaped. They are narrow and they are ribbon shaped. These submerged leaves are narrow. Okay, aerial leaves are broader and submerged leaves which are present in the water, they are narrow. Okay, so here if you take, why the difference in the shape of the structure of the leaves? It's because of environment. That's why it's called environmental heterophily. What is the best example for this? Buttercup. Now you come back to the definition. What is the definition? Plasticity, process of formation of different structures. In young plant you can see a different structure. When plant becomes adult you can see a different structure. It's because of the changes whatever happens in the plant. And in response to environment, maybe sometimes the different structures are formed in response to the environment. And they form different pathways. It's called as what? Plasticity. I repeat again, development, if you look at, one of the developmental process is plasticity. What is the plasticity? I repeat, process of formation of different structures in response to environment and form different pathways. That is the definition for what we call plasticity. Now, what are plasticity? You can take one more example for plasticity is heterophily. Hetero, different structures. Phily means leaf. A different structure of leaves. Where do you find? You find in case of cotton, coriander and larkspur. So, cotton you know ricinus, right? And coriander you know coriander sativum, larkspur. These are the three plants. What you find here is heterophily condition. Now, in this heterophily condition, in these three plants you can see a different structure of leaves. Hetero means different structure, phily means leaves. You can see different structure of leaves. So, but for example, you can take coriander. Coriander you can take young plant. Young plant having the two small lobes here in young plant. It's very simple structure. But when young plant converts into an adult plant, you can see on the same, you can see 4 leaves. I mean one leaf has 2 lobes. When it becomes an adult leaf, it has many lobes. It has many lobes. So, here the coriander is the best example which shows what is called heterophily, formation of different structures within the leaf, and that is heterophily is a good example for plasticity. Plasticity is a good example for one of the developmental process. Then here sometimes you call environmental heterophily in some plants. Why do we call environmental heterophily in some plants here? For example, you can take buttercup. So, what happens in buttercup? You know, some leaves are aerial, some leaves are submerged in water. So, aerial leaves are broader and submerged leaves are ribbon-like and narrow. This difference is because of changes in the environment. Leaves are broader in aerial or in air, leaves are narrow in water. This is example for what? Environmental heterophily. The factors influence the development. The factors which influence the phenomena called development. They are divided into 2 categories: intrinsic factors or internal factors. Extrinsic factors or external factors. Intrinsic factors or internal factors. What are intrinsic or internal factors? So, if you take intrinsic or internal factors are further divided into 2 categories, classified into 2 groups or 2 types, classified into 2 types. What are they? We can say intra-specific factors and they are interspecific factors. Okay, internal factors, intrinsic factor further divided into 2 types. What are they? Intra-specific factors, inter-specific factor. Intra means within the cell. Intra means within the cell. Intra means what will come? Genes. Genetic. Genetic means it includes genes. Genes will decide within the intra means within the cell. Within the cell, genes will decide what type of development process has to take place. Intra means within the cell. Within the cell, there are specific factors. Which factors? Genetic, genes. Genes will decide what development process has to take place in the organism. That is called intraspecific factors. Then what is interspecific factors? What is interspecific factor? Interspecific factor is means they produce in between two cells, in between one cell and one cell. So this we say, this is within the cell, you find nucleus, nucleus contain DNA, on DNA who is present? Genes are present, genes will decide. Coming to interspecific factors, this is one cell, this is another cell, in between what you find? Plant growth hormones. Plant growth hormones. What are plant growth hormones? Or we say plant growth regulators like auxins, gibberellins, cytokinins. Okay. What are interspecific factors? In between two cells, who will find? Plant growth regulators. Some chemicals are released. What chemicals are they? They control the development processes. In between two cells, in between two cells, in between two cells, there are some chemicals. These chemicals, they involve in developmental processes. What chemicals they are? They are called plant growth hormones, plant growth regulators. What are they? Auxins, gibberellins and cytokinins. If they play an important role in development process, they are called interspecific factors. Coming to extrinsic or external factors, this you know. What are they? You can take light, temperature, gravity, right, and along with that minerals or nutrients, including water. What are external factors or extrinsic factors? Externally, the environmental factors also play an important role on some of the developmental processes. What are the external factors or extrinsic factors? You know, surrounding environment have the influence of light on the process, temperature, gravity, minerals or nutrients and on water. So these factors also influence development and these factors also influence development. So development has both internal factors, external factors. I repeat, internal factors are intraspecific or interspecific. What is intra? Within the cell. Within the cell, who controls the process? Genes. Interspecific, in between two cells, there are some chemicals. These chemicals are auxins, gibberellins, cytokinins. They are called plant growth hormones. They also decide developmental processes. And what are external factors? Light, temperature, gravity, minerals or nutrients and finally the water.